always look at science from the point of view that we should always be testing it. We should always be thinking the null hypothesis could still be right. Even if the data is very convincing, it's still a possibility. Never talk about the science, obviously, because it's not settled. Science is asking the question, is this still true? Is this still true?
Today I sit down with Dr. Monty Gill, a lecturer at the University of Guelph-Humber, one of the finest research scientists that I've known, and my favorite person to talk science with. He completed a PhD in molecular genetics at the University of Toronto, followed by postdoctoral fellowships at the Amgen Research Institute, Ontario Institute for Cancer Research, and the Lunenfeld Research Institute. Dr. Gill then transitioned into a research associate role at York University, where I had the pleasure of working with him and learning from him. As a scientist, he focused on several signaling pathways, including the regulation of the retinoblastoma gene, telomerase activity control, RNA interference and insulin signaling, and AKT targeting of cell cycle proteins. His respect and mastery of the scientific method is matched by few, and is built on a foundation of tens of thousands of hours spent in molecular biology labs, resulting in over 20 publications to his name. Today, he lectures in the kinesiology department at the University of Guelph-Humber, where he continues to impart his endless well of knowledge on up-and-coming scientists. And without further ado, my good friend, Dr. Monty Gill. Now let me start off with this. Let me see if you agree or disagree with this statement. The most important advance or discovery in the history of biotechnology are restriction enzymes in the nucleases.
Well, I think because my career goes back a little farther than yours, at the beginning of my career, we were still finalizing and confirming the structure of DNA and the fact that the order of the nucleotides actually coded for the sequence of a gene and therefore the order of the amino acids and proteins. So for me, that's even bigger. Since then, restriction enzymes, PCR maybe?
Yes.
Yeah.
So, but without restriction enzymes, you couldn't do any of the cloning that you did a lot of in your career, in my career, or any of the genetic sequencing.
Yep.
Yeah. So in my opinion, that is, so you would say for you, the most important advance is data-driven in the sense that we know what the structures are, we know the order of amino acids, we know the nucleotides that code for them. It's more of a data thing rather than
I think you need the knowledge because, well, the restriction enzymes are awesome tools, as is a PCR machine, as is a automated DNA sequencer. If you don't have the knowledge, what are you going to do with all these tools?
That's true. Yeah. So you would say that in your career, or maybe not, let me ask you, in the course of your career, what is the most exciting advance that you've seen? In all of your career, it's been a long career.
I think that it would be PCR because before that point, so when I started my very first lab, we were trying to isolate what we thought was going to be a recessive oncogene that was causing a type of cancer. And the way we were doing it was so primitive. We were basically using restriction enzyme and cutting up an entire cell's worth of DNA and cloning all those massive fragments of DNA and trying to find one that had potentially the gene on it by doing hybridization of radioactive probes onto these blots that we took off, these plates of all these colonies of viruses. It was just, we had about 20 people working on it for 5 years. Now you would just go, oh, I look in the database, oh, that's the sequence for the open reading frame that we think codes for this particular gene, designed to oligonucleotides, order them, they show up the next day, put in the PCR machine that afternoon, you've got the piece of DNA. So
it's amazing. It's amazing how quickly we take these things for granted.
Yeah.
Right? We just look it up, PubMed, that information is right there.
Yeah, but the sequencing of the whole genome, right? Yeah, that's also a massive advance.
Yes. So maybe you can give us a little bit of a insight into your journey as a research scientist in your early career. Now you're a professor, but how did you get into wanting to do research in molecular genetics, which is a big part of biotechnology? How did you get into that journey?
I think when I was maybe back in early high school, I got interested in science. I just found the idea of the scientific method. It just was so logical to me. And that's, I guess, the topic of mostly today's discussion is the scientific method. It just made so much sense. And so I wanted to get into science. Well, what aspect of science? Physics or chemistry. Right. And biology was the most interesting because, again, that was around the time when we were going, oh, that's the structure of DNA, the double helix, triplet code for nucleotides. It seemed like that was going to be, and I think it has turned out to be, just a massively expanding field of science. So I was interested in that. And more specifically, what kind of led me into my first lab, my first graduate work was cancer, because back then in the 70s and 80s, cancer was just devastating. Like it was killing people 70-80% when you got cancer, and everybody's family was impacted by cancer, of course, including my own. So I was able to go, I'd love to work on how cancer works and maybe make some progress towards making it less deadly. And it's in this fascinating field of molecular biology. So that's how I ended up in the field.
Right on. And you mentioned that you were really interested in the scientific method, which is at the core of all research science, applied practical science. Maybe we should define it for our students. So scientific method: observation, hypothesis, then you experiment, you collect data, you analyze it, you come to a conclusion, you publish this, you go through a peer review process, and then you replicate it if you need to. Somebody else can replicate your work to confirm it or not confirm it. And then you've got a theory or a law at the end of the day. So this is maybe you can give us some insight into how this sort of process works into the everyday life of a research scientist.
Right.
Where does it? Are you keeping that in the back of your head as you're doing science? Are you thinking 10 steps ahead? Well, it's not really 10, but a couple of steps ahead.
Well, you are always thinking multiple steps ahead, because so the way you described it, that's perfect. So we make observations about the world around us. Going probably back to that first exciting research that I got involved in, which was trying to find this recessive oncogene that was causing a particular type of cancer. You observe that there seems to be a Mendelian inheritance of this cancer. It seems to be behaving like a recessive inherited trait. So we thought, well, it seems like there's a gene that if it's mutated in both copies, you lose the suppressive effect on growth, and a cell grows uncontrolled and it would cause cancer. So that's like an observation we're making in the real world, observing the inherited pattern of this cancer. All right, design a hypothesis. Well, we think there's a recessive oncogene. Okay, so now you got to start designing experiments. You got to try to find the gene. So you start off by going, well, I would hypothesize if there's a recessive oncogene that we're going to see if we take the cancers, the cells from cancers, from a number of different patients that have this tumor, that we're going to tend to see deletions that we can see at the chromosomal level in one particular area fairly often. Now, sometimes there'll be point mutations, but often they're big gross deletions. You can see them by just looking at the chromosomes. And so then if you do that, we started to go, well, it looks like this 14.3 region of chromosome 14. This might be the area. So we started looking in there, trying to find a gene that may be responsible. So isolate a number of candidate genes, and you hypothesize, well, if one of these is the actual gene that's damaged in these retinoblastoma cancers, if I was to, for example, put that back into a cancerous retinoblastoma cell line, it might reverse the phenotype. It might slow its growth. If I was to knock that gene out in a mouse, should they start developing tumors? So you design these experiments to test the hypothesis, and you never know what you're going to get, because we got to be aware of the null hypothesis, of course, which
you
is,
took the words right out of my mouth. Null hypothesis.
Yeah. You always have to say, well, there's my hypothesis, or our hypothesis, or the hypothesis, that this is an explanation which we're putting forward that might explain the phenomena we're observing. But there also should be the alternate, the null hypothesis, that we are incorrect. For example, maybe there isn't a recessive oncogene causing that cancer. Maybe this particular gene is not the one. Or if we're trying to explain how this gene is actually functioning, well, our hypothesis might be incorrect. And so we always have to be prepared to interpret the data. It either supports the hypothesis or it's the null hypothesis. A really common lack of use is the null hypothesis is when you look at data and you have to do, of course, statistics on it. What's the average? What's the standard deviation? There's always the null hypothesis that a difference you're seeing might just be random chance, random data that came out that day. And you have to therefore always be aware that there's a chance that what you're seeing is just random data that isn't real. So increase the number of samples
and set your p-values accordingly.
Set your p-values accordingly. And I think people forget about the null hypothesis and they start searching for data that supports the hypothesis. Set that data aside. It doesn't support the hypothesis. Well, it's actually supporting the null hypothesis, which is that you are incorrect.
And I think along those lines, a lot of green students will overlook, and we see this in our lab sometimes, is you'll overlook a negative control. You have to know, you have to know what a negative result looks like, and therefore you kind of set up a control which forces a negative result so that you can then use that to decide if your null hypothesis is correct or incorrect, or which way are you going. Yeah.
That's massive, actually. Just to expand on that.
Yeah.
You need to devote as much energy and time to thinking of explanations for how the phenomena might be occurring, the hypothesis.
That's right.
You have to spend just as much time thinking of things that might be causing the result to occur that isn't your hypothesis. And that's where you come up with all the various negative controls. Don't forget to spend time thinking, well, if it was this thing, which isn't the hypothesis, that might be something that's causing the result. So make sure you try to design a control that might point in that direction.
Yes, exactly. Yeah. Often overlooked, but something that's very important. I like to stress that on our students. Now, you've talked about the gathering of data and doing experiments. I find one of the most common questions that I get from green students who are thinking about doing research is, on a day-to-day, how do you decide which experiments to run? If you were to observe a research lab, from the outside, and you'd see these people working, you'd be in awe of the experiments that they're doing, and you'd wonder. A lot of students wonder, how do they decide to do that experiment or that experiment? So does somebody tell you? Does your boss tell you? Do you have some autonomy? Can you do it yourself? How does that work?
Well, I hate to get into the judge part of science, but I think your students should be aware, especially if somebody's considering a career in science. A lot of times what decides what experiment you're doing today is you're doing the experiment that you did yesterday that failed.
Right.
Because of various reasons, you made a mistake. There's a lot of repetition. Plus, you're often repeating experiments because you need more sample size. Yes. So a lot of times you're just doing the same experiment, either to get it to work or to get more data. Second, you're often thinking of new experiments. And where do those come from? Well, it comes from a lot of places. Sometimes you're lying at home trying to fall asleep and you're trying to figure out, how am I ever going to be able to convince someone that in fact this gene is a recessive oncogene? And it might come to you. Or you're sitting over coffee with somebody in your lab and you're talking about your data, and somebody will say something and you'll come up with the idea of the two of you. Or sometimes it is actually your supervisor, who is pretty smart usually in the field. That's why you chose to go work with them. And sometimes they do bring ideas for experiments to you. Although my experience has been, it's actually is come up with organically a lot more often than directed.
Yes.
But not to say your supervisor doesn't come and say, I think you need to try this.
Right, there are different approaches, that's a great idea. Hands-on, hands-off approaches. Yeah. At the end of the day, you're there because you're good at what you do. Yeah. So there's a lot of trust being put into you.
That's right.
So another part of the scientific method that students don't really see until they decide to do a postgrad or graduate work is the publishing part and the review part. So what is that like? After you've collected data, you've analyzed it, you think you've got some sort of result, a conclusion, where do you go from there?
That's a part of science that's become more and more difficult as time's gone on. When I started as a PhD student, there was probably about 1/10 the number of journals as there are now. Now, I'm not saying that there's 10 times as many journals and they're all fantastic quality. The high-level journals are still the high-level journals, but they're very hard to publish in. And it's always your goal to publish in the best journal you can. You get the highest readership. Yeah, people realize it's an impact paper a lot of times based on the journal it's published in. So the problem is you come up with a bunch of experiments that convinces you and your supervisor and the other people in your lab. Then you submit it to that journal. Well, the first thing they do is they send it out to 3 experts, peer reviewers. And those people are experts in the field and they are tough. Right. They generally want to rip your paper to shreds, say that every one of your experiments doesn't prove anything, and they look for faults. And then they suggest an entire brand new set of experiments that you would need to do to form a convincing argument. So a lot of times, after you do all that work, write the paper, submit it, you get back a brand new 2 years worth of work to do before you're going to convince them to accept this paper for publication. Right. That's a big part. Publication ends up being as much work as the original work that you conceive of and execute. Right. As we were talking earlier, we were saying that there's no other industry where a year can fly by in the blink of an eye. Just when you think you're done, they pull you back in. That's right. And they make you do more experiments. That's right. We've been there. Yeah. Now, so you get peer review, get published. At the end of the day, somebody will try to replicate it if it's interesting enough. There are tons of articles that are published that no one ever reads. Unfortunately, they may or may not be replicated. But at least in the high impact stuff, the interesting lines of research, somebody's trying to replicate. So there are labs that focus on doing that. It's in their best interest. You might have a publication which maybe rebuts your publication. Absolutely. And then you kind of start this back and forth war where you're trying to
replicate their stuff, and they're trying to replicate your stuff. Right now, we see that with the room temperature superconductor, the LK99. People are trying to replicate that right now. May or may not be a real thing. That's besides the point. But could you maybe tell us about a fascinating or interesting challenge a research project that you worked on where the scientific method played a very crucial role? I think one of the stories you once told me was about your retinoblastoma, or no, rather the mTOR project that you were working on. Yeah.
That's actually process. So that, I mean, you're always following the scientific process, the scientific method in any research that you're doing.
Absolutely.
But what we're talking about here more is how the science really works.
Right.
And this is an unfortunate experience, I guess, that I had. So this would be my second postdoc. So I did a master's project on retinoblastoma. I did a PhD on E2F and retinoblastoma and their role in cellular differentiation. Then I worked on telomerase, and then I worked on the insulin AKT signaling pathway. So my project was to use a new technique that had come up called RNA interference, which is a way to specifically transiently knock down the expression of any gene you want with antisense RNA. And so I was targeting every part of the pathway. There was like 11 different members of the pathway. And the result I was getting was that when I knocked down the TOR kinase, which is the kinase that phosphorylates AKT, which is a really important kinase. Well, what I was getting was it was resulting in a decrease in PIP2 to PIP3 phosphorylation, which is farther up the signaling pathway, according to the settled science.
Right, the settled science said that mTOR was unidirectional.
mTOR was downstream. There was PIP3 was produced, then something else happened, then mTOR was activating AKT. But when I would knock down mTOR, I would get a reduction in PIP3, which that doesn't make any sense. It's not the kinase of the PIP2 to PIP3. So I investigated it further. I purified TOR, and I had purified PIP2, and I put them in an in vitro assay in a test tube, and the TOR kinase was phosphorylating the PIP2. Well, that's incorrect according to the literature. TOR doesn't phosphorylate PIP2, but that's what I was getting. And so when I would present this data in my own lab to my supervisor, to the institute as a whole, I would go to conferences. It was not accepted. There was a lot of controversy, a lot of basically standing up and yelling that this is stupid, you're wrong, I don't know how you're getting this result, it's wrong. To the end where every experiment I did, though, I kept getting the same result. And so in a sense, I realized this is never going to be publishable. I've spent 5 years of my life. This is going nowhere. And it actually made me leave science for a while because there was nowhere to go. So sometimes the way science works in the real world, which is it's very difficult to contradict the agreed upon narrative on how something works. And as we realize, though, over the many years of science, sometimes that narrative is wrong. But when you're the first person to try and contradict it, it can be very difficult.
And this is a terminology we hear nowadays a lot, the science. And we'll get to that that idea shortly.
Okay.
So how do you address uncertainties or things that deviate from the science in your research? So you're the person that comes across something that was totally unexpected, but you're convinced. What about the scientific method helps you tease that out, whether it's true or not?
Yeah, well, that's always your goal. That's always the exciting thing for a scientist, is that you get a result that says, hey, this is something new that people don't know.
That's what we're in it for.
That's what we're in it for. So you pursue it like mad because that's what you're excited about. So you're following the scientific method. Now, maybe it was experiments you were doing for this reason over here, but you're looking at the data you're getting. You're looking at the results that are happening in the real world. You're looking at the natural world and going, I'm observing these things. What would explain them? So then you would design a hypothesis that might explain that, design some experiments to test it, and that's how you come up with some new experiments to do for the next year of your life. So you're always following that scientific method, trying to come up with an explanation and designing experiments to test that explanation for any data that you're producing, especially if it's new, interesting, and exciting.
Now, how important, we talked briefly about replication outside of your lab, somebody else replicating your work. How important is it in your day-to-day work to replicate a result that you came across yesterday? How important is it to replicate it? A couple
Yeah.
times, never once, many times?
I think it's super important to replicate it because generally when you see a result that you're interested enough in that you may, for example, want to build some new experiments on top of that, you may take that result again, observing results in the natural world, somebody's published paper, and you want to then design some new experiments to test the hypothesis. You're building on top of that work. Well, you better make sure that you believe that data. So that's a main reason why I would often replicate something, because I wanted to build on top of it. And I didn't want to build on a shaky foundation. So I'd want to confirm it, not just to confirm it, because honestly, you can't publish confirmation papers.
Right.
Nobody publishes the exact same experiments done by somebody else. They get done in the process, I suppose, of doing some new work. So you're usually doing it to build on top of that work. And it is critically important. And eventually enough people, you're at a conference, you're talking, you know, that new result from last year, none of us could replicate it.
Right.
Could you replicate it? No. Could you replicate it? No. Or yeah, sure. It's real.
Right.
So that's often how it would work. It would happen more just over essentially scientific discussion more than actually publishing a confirmation paper.
Okay. Now, when I say the term the science or trust the science, what's the first thing you think?
I get really upset. That's a crazy idea.
Why?
The science, what the science is, as somebody who is a scientist, and I was really frustrated listening to a lot of people who aren't scientists tell me, a scientist, about how science works. Science isn't an agreed upon set of facts. Science is challenging the hypothesis constantly testing the null hypothesis, because just because you had a hypothesis, you designed some experiments, or someone else did, and those results from those experiments supported the hypothesis, doesn't mean the hypothesis is confirmed. It means to this point, we haven't nullified that hypothesis. Right. So you should constantly be challenging the accepted hypothesis explaining a set of events in the natural world, because a lot of times it turns out that our explanation wasn't correct. Newtonian physics being replaced by Einsteinian physics.
I was just going to say, had we trusted science, we'd still be at relativity and no quantum physics, chemistry, nothing.
Exactly.
The
so
bread and pudding method or analogy would still be in play.
Yeah. The science is actually challenging the accepted facts, not accepting the accepted facts. That's religion.
Right. So, okay. So along those lines then, could you give our students some advice or some sort of, I would say advice for anybody who's interested in doing research science? What makes a good research scientist? Some advice.
Okay. I do this a lot actually with my students as well, students that are considering a career in research. It's a really challenging career path. Honestly, I don't want to discourage anyone, but if I'm being honest, I'll describe that what you need to be a successful research scientist is, number one, you need a good hypothesis. And by that, I kind of mean a correct hypothesis, or at least the data is going to support that hypothesis. If the hypothesis you start working on in a lab as your project is incorrect, you're going to spend 2 or 3 years to go, okay, that's a bunch of negative data. So you need a good hypothesis. You need a good environment to work in. So that means a good supervisor, a good professor that has a good lab with a lot of funding, a lot of equipment and materials so that you can actually perform all the experiments you think of. And then you need to have a couple of other things. You got to have good scientific writing skills, because it does you no good if you don't publish those papers. So you have to be good at that. Of course, you have to have a good work ethic, because working in the lab, it's a lot of hours, a lot of experiments, a lot of time you put in. They fail for reasons beyond your control. So a lot of time. So now you've done all that. You've successfully published on that research project. Well, that was your master's. Then you do it again on a different project in a PhD in a different lab. Then you do it again on a postdoctoral fellowship in yet another new lab, and probably a second one. Then you've been successful at all those stages. You had good hypotheses, good research environments, lots of funding and support. Then you have to try to get a position where you are a principal investigator, a tenure track professor at a university. Then you got to get grants, and then you have to design a whole new set of projects and have them come to fruition. And then you put yourself up for tenure, and hopefully your university gives it to you. Now you've made it.
Right.
That's a lot.
Yes,
that's
a lot. Excellent time management skills. Absolutely. Now, one thing that I learned, so we met when I was doing my master's. You were a research scientist associate in the lab that I was doing my master's and then PhD in. I learned a lot from you. One of the biggest things, most important things I learned from you is resourcefulness. The best scientists that I saw when I was going through my education were those that were resourceful. You may not have the funds, you may not have the proper tool, proper equipment to finish the experiment, but if you're resourceful enough, you can get through it. Would you agree?
That's right. There's a lot of collaboration that goes on. Sometimes, again, as you mentioned, you don't have the equipment, but somebody else down the hall does. Now, as scientists, we're a little jealous of our idea and our great hypothesis that seems to be proving out, but we need to do this experiment and we're not able to do it. Maybe we don't have the technical skills, or maybe it's just simply the equipment. And then we have to do the thing that we'd rather not do, bring some more people into the project and share the credit.
Right, dilute your name down a little bit. Yeah.
A lot of times you have to do that. I'll give you a great example. You're working on a brand new protein, as we did in our research project that we worked together on. And it would have been really nice, for example, to have an antibody against that new protein, because one didn't exist, of course. It was an uncharacterized gene. Well, we weren't able to produce that antibody ourselves. We contracted that out to a company. It would have been really nice to have the three-dimensional structure of the protein. Well, we weren't going to be able to do X-ray crystallography. We might have had to bring in collaborators that had those technical skills and equipment. So collaboration is a lot of what you do when you're being resourceful. But sometimes it's also just finding a way to do it with the equipment and materials you have on hand. Right. Thinking of alternate experiments that might also accomplish the same goal that you can do.
Can you tell us of a memorable or transformative moment in your career that really cemented your passion for research?
I had a couple, I think, and it's kind of the same story. It's when you have a hypothesis and then you get that first result that, oh my God, this hypothesis might actually be correct. And my first example, it was my very first project. So when I came into the lab that was trying to find the gene that was responsible for this retinoblastoma cancer, we had the candidate by the time I got there. The people had been working on it for about 5 years. We had a candidate, but we weren't sure. So my project was going to be, I was just a young master's student. I was just one little person over in the corner in a big lab. My project was going to be, is this gene, the retinoblastoma gene, is it regulated? Because at the time, everybody believed in the literature, it was essentially a housekeeping gene. It was always expressed at the same level. It was constantly on. There was no regulation in expression. Maybe just its activity was modulated farther downstream, like the protein was modified or interacted with another protein. But we didn't believe that it was expressed at differential levels. So my job was to go look at the promoter, the transcription of the thing. And my hypothesis was that it was regulated. When I looked at the sequence, I said, that looks like an E2F site. So first thing I did was I said, if that's an E2F site, E2F is a transcription factor that's highly active at a particular point in the cell cycle. So I synchronized some cells at one spot in the cell cycle, then I let them go through and I took samples and I looked at the levels of the retinoblastoma messenger RNA. And I got the result that it was really low in G1, and as we transitioned into S phase, it spiked. So that totally fit my hypothesis that, number one, it was regulated, and it also confirmed to that point my hypothesis that it was regulated by the E2F transcription factor. And that was about 30 years ago, and that result still holds. It could be disproven at some point, but that result still holds. That was super exciting. That was my very first hypothesis as a 19-year-old graduate student. It was very exciting.
And ultimately proved to be correct?
To this point.
To this point.
We always have to say to ourselves, it's never settled science.
So that's an interesting aspect of research, is you might come across something exceptional, something new and novel, revolutionary, but maybe only for 10 years. Years, and then all of a sudden you're not that interesting person anymore. Or, you know, there are egos involved. This is one thing a lot of people outside of science don't see or recognize, the egos involved.
See, it's in my experience, which was fairly long, about 30, 35 years in a lab, when you're dropping a new idea into a pool that doesn't have a formed opinion yet, it's much easier to publish that and set the narrative that, for example, the retinoblastoma gene is transcriptionally regulated by the E2F transcription factor. But when you're contradicting something that's pretty well established, that's much harder, because again, who are they going to send it out for review to? The people that publish the work that's already. And that's the problem. And that's why you'll often hear the idea that science only moves on when the old scientists move on.
Mm-hmm.
Once they're gone, they're no longer
Right.
reviewing the papers, and they no longer enforce the narrative.
Right.
Not saying the narrative is wrong all the time, but it doesn't matter. They have a vested interest in the narrative not being disproven. Their whole careers are built on it.
So correct me if I'm wrong. You didn't do any clinical research. You never worked with humans. But
No, human cell lines.
human cell lines.
Yeah.
But that notwithstanding, there are ethical considerations that have to be done. So how do you pursue, or I should say, how do you navigate and balance the pursuit of scientific data or knowledge or curiosity with ethical considerations?
Honestly, that's a tough one.
Right, and those ethical considerations can be anything from should we do this experiment to how do we cherry pick this data, or do we cherry pick this data, right?
On the do the experiment side, that's so hard, because when you're pursuing the truth, the confirmation of your hypothesis, new information on how the world works, you're so excited. This is what you do. You want to do that experiment. You don't want to sit back and go, I don't think I should do that experiment. It's very difficult to not do it. I'll give you an example. When I was often working, I was genetically engineering viruses to put in cancer-causing genes because I wanted to express them in cells and test some hypotheses. So I was making these viruses that infected human cells that expressed cancer genes. Those were obviously very dangerous viruses, and probably mostly dangerous to me. They were replication deficient viruses, but I had large amounts of purified ones. I could have infected myself.
Sure.
So ethically, should I have been doing those experiments? That was very dangerous, but I wanted to do those experiments. So honestly, it's very hard to enforce ethical issues yourself,
right?
Because you want to pursue. Another example is working with lab animals,
right?
Working with mice. To be honest, I was really lucky. I kind of managed to avoid working with mice my whole career, even though at times I was kind of being pushed in that direction because I was very uncomfortable around that.
Mm-hmm.
But obviously a lot of scientists do it. It's like what you need, I hate to say this, but you need somebody from outside enforcing whether something should be done ethically, because from inside, it's very hard to enforce those restrictions yourself.
And I would imagine at this point, all research institutes, universities, colleges have ethics boards.
Every grant has to go through
that
ethical.
help you answer these questions.
Yeah.
Help you decide.
I do think sometimes, though, there is a certain amount of knowing that the ethical board is looking at your grant application, and you may not necessarily fully describe an experiment that you're trying to not draw attention. There's ethical issues everywhere. Your other ethical issue was the one about
data.
About data.
That's right.
And that's huge, obviously,
Yeah.
because in the end, you're not moving on in your career, or you're not getting your grant application renewed, or you're not getting your tenure at your university if you're not publishing. So while you, of course, ethically, you would never want to not tell the truth, there's extreme pressure to publish. So I do think it happens that a lot of scientists are tempted to cherry pick data. I wouldn't go as far. I can't say I've ever seen anybody manufacture data, but obviously there's a few issues in the news right now with that happening, with Alzheimer's research, for example. There is temptation. There's obviously incentive to do it. And not every scientist is a saint. Not everyone holds themselves to the same ethical standards. So I think we have to be aware that that's always a possibility when we're looking at scientific data. You always have to consider the possibility that the data is not.
And now we come full circle. Scientific method, you have to replicate those results. So if you're looking at some publication where you think, this doesn't make sense, could be cherry picked, you replicate it. And now we see things like the Alzheimer's studies.
How long did that go on? How long did papers
30 years,
confirm that for 30 years? And it was actually manipulated scans, manipulated pictures.
That's right. There was also another issue in the news as well. It was the Alzheimer's, and now it's slipping my mind. I think it was something to do with sugar and fat metabolism, but that's for another day. What excites you the most about the future of biotechnology, molecular genetics, molecular biology?
I'm probably a little bit colored by the fact that, like, for the last 10 years, I haven't been doing research. I've been teaching a lot of biochemistry and metabolic pathways. And also, I'm getting up in years. So obviously it's an interest of mine using the molecular biology advances in our knowledge to live longer and live healthier. And I think that's the thing that excites me the most, the idea of being a healthy, active, productive 80-year-old person as opposed to somebody, confined to a walker in some home somewhere. And I do think the science is giving us major advances in that. And I try to, I'm sure that many of your students have heard the term biohacking and they're curious about some things. I do think that there is some biohacking that you can do. Maybe if it's just eating the right foods as a really basic part.
Right.
But I do believe that you can have a major effect on your longevity and health with all this advance in biological knowledge.
Right, especially when you take into consideration personalized medicine, personalized science. You can get a lot of information that pertains specifically to you and then adjust your eating habits, for example.
That's right.
Right. Okay, as we wrap up here, Monty, if you may, some words of wisdom in terms of the scientific method and our students. They should go away thinking about the scientific method. How?
I think they should always look at science from the point of view of that's kind of what we think at this point. They should never look at it as in that's known. They should never look, even if it's in a textbook, that doesn't mean that it's tablets from God. Those are the current state of what we think. There may be a lot, there may be a medium amount, there may only be a small amount of data to support it. And we may have new data in the future. We may have a scientific breakthrough that changes our view on things. Always look at science from the point of view that we should always be testing it. We should always be thinking the null hypothesis could still be right. Even if the data is very convincing, it's still a possibility. Never talk about the science, obviously, because it's not settled. Science is asking the question, is this still true? Is this still true?
Thank you, Monty.
Thank you.